such a pleasure to welcome you all to Preservation Week and to celebrate collections, collaborations, and our wonderful, wonderful staff. So, as Jake noted, I will be talking about fragments discovery and creating knowledge. And this came out of a discussion we had with some of our curatorial collections. What I wanted to do is start by just sharing some of our staff. This is not all of our staff, but some of the staff who were working on this project along with Mariana Stell from Rare Books and Special Collections. So on a number of projects, we've been starting to think about how we can sort of learn more from our collections. And in discussions with Stephanie Stylo and Mariana Stell from Rare Books and Special Collections Division, we realized that they had boxes of fragments, glorious fragments, that were in the process of being worked through. So as Mariana shared with us, part of the work with this is identifying the text, creating minimum level catalogue records, and then putting them into the queue for digitization so that scholars can access them. One of the things that's really interesting for us is that the technical analysis and the characterization of the materials, which will help aid provenance and potentially reconnect the collection items, also allows us to learn a lot more from the collection themselves. And you'll see as we go through just how we're starting to build the knowledge. I will note that this is a project in its early stages, and so I hope that sometime in the future, some of the staff working on this will be able to share more as we're learning. So as you can see here, there's some wonderful parchment fragments that we're working with. Lots of different texts, whether we're talking Latin or Italian, and you can see they're in various stages of, some potentially may have seen better days. They've been reused in various ways. Other ones here, you're starting to see a lot of staining. Strangely, in preservation research and testing, we love stains. We're very interested in what those are, because the more that we can do with our techniques to understand historically what has happened to these materials helps us aid the preservation and work with our collections care, curatorial and conservation staff. And another one here, you can see just wonderful little fragments that really delightful and have little hidden gems in all of them, as you'll start to see. So I wanted to introduce, for those of you who aren't familiar with that, the concept of fragmentology. This is really a guess. There's more than 30,000 fragments of manuscripts in both North American and other worldwide collections, but often the fragments historically hadn't really been deemed worthy by scholars, worthy of study. But this is changing a lot, particularly as we see certain of these fragments being digitized, and as they're out there, scholars start to realize this potential fragment might have come out of this volume and got separated at some point in its history. So there's just a huge source of knowledge within these fragments that is largely untapped, because if they're not digitized or not accessible, then people aren't aware of them or using them. So the approach that we'll be taking, as you'll see as I go through my presentation, is to add and explore information to the curatorial knowledge and try to understand a lot more about both these unique fragments, but also how they can help us hone and advance some of the techniques that we're using. The technique that we take is a materials analytical approach, where we start with imaging across the entire object, not just in one spot, and then all of the complementary analytical techniques build knowledge as we go through that. I'm only going to talk about a few of the techniques we have. There are many more, and as Jake said, hopefully with ALA you'll get to meet more of our staff if you're in DC and see some of the other techniques as well. But each of these techniques works on a different aspect of knowledge about whether it's the substrate, the parchment, or the inks and pigments, and potentially how we understand how they might be degrading and why some are in better condition than others. So what we have, for want of a better term, we affectionately call this the GO team, which is a group of staff which changes depending on the project. And we start, as I said, with the imaging and work through these other techniques. Here you're seeing a range of different staff, in fact, working with Stephanie Stylo in the second image there in the Rosenwald collection area. So what do we do when we learn from the fragments? Well, we can start to characterize and identify the materials, which is the substrate, so the parchment in this case, the inks, the pigments, and any stains or discoloration. And if there are ones that are particularly challenging, we build our techniques to figure out how we can actually identify those. We want to use that information to understand why it's in the condition it's in. And through understanding that, can we then learn more about how to stabilize and how to preserve it? Can we also image through the stains and find original text, which helps scholars learn more about the provenance? And then also, what information can each of those techniques that we're using give us so that we can build upon our knowledge and expand techniques or instrumentation that might add to the information and data we can gather and share. I briefly wanted to mention our reference collection, CHARM, which, as you can see here, has lots of different materials. For this particular project, we're more interested in the parchment, and so CHARM stands for the Centre for Heritage Analytical Reference Research Materials. Here you can see a number of the different types of parchment skins we have: goat, deer, calf, and sheep, in different levels of processing. So with that, we can understand how potentially the original construction or creation of that parchment might actually have led to something being in better condition or not so. We also have staff, and this huge thanks to Cindy Connolly-Ryan here, who have recreated historic recipes. And as you can see here, iron gall inks made from soluble tannins that are extracted from gall nuts or bark have been mixed with different vitriols. And those are important, as you'll start to see when we talk about one of the techniques. We've created a number of different reference sample sheets that we can do different things to and have different combinations of binders and different percentages. All of these allow us to then non-invasively use the techniques that I'm going to talk about to better understand what's in those materials, what's in those fragments, what's in our collection. We start, before we do any analyses at all, working with in this case, the curatorial staff and all the preservation staff, to look at what might be potential regions of interest, how we start to answer the questions. This was just a preliminary start point. And for this one, we really wanted to look at the clean parchment, the darkened or potentially damaged parchment, and different centers of the regions across this, and potentially the inks. So what we do is we identify these areas, and then for each of the techniques, we look at these particular areas with different instrumentation to build up that knowledge, to essentially do document archaeology as manuscript archaeology as we work through. So I'm going to start with multispectral imaging. I know many of you already know what this is, but it's a non-invasive technique that captures information both in and out of the visible region. So we start from the ultraviolet, move through the visible and into the infrared. And all of those wave bands will capture different information from the fragments because the composition of the parchment or the pigments or the inks respond differently based on their composition. So with this, we start with what we call full spectrum color images. The beauty of this technique is we have more wavebands than you find in a normal RGB camera. So that allows us to do a really intense, very precise rendering of what the actual color of that material looks like. Excuse me. So you can see here a composition of that on the left. As we start to add the different modes of illumination, the first one we do is reflected imaging. And you can see from the image on the right, as we rotate through different wavebands, different materials respond to different wavebands. And so that allows us to start to firstly understand which ones, if there are two blues, for example, that look the same, are in fact the same or not, and then how we actually add knowledge with the other techniques. We can also do low angle raking, and you can see here that in fact, a piece of parchment is not 2D, it's in fact 3D. And this helps us understand what might have happened to different regions. And as we build the knowledge with other techniques, we add and understand why these areas might be flattened and others are cockled. Transmitted light, which is light coming through from the bottom of underneath the object and captured through the lens there, allows us to look at some of the construction of that and or stains or light and dark regions. This in fact is a paper document, but you can see for paper, for example, we can pull up a rendering of the watermark. Of course, we don't find that in parchment, and then some of the changes that have been made. Also fluorescent imaging, different materials fluoresce, so we can actually use that to help identify different varnishes or oils or glues. In this fragment, it was actually reused as a binder waste and repurposed as the cover of a book. So we're starting to see some of the glue. And then, of course, with other techniques, we can add and identify what that might be. What I do want to make very clear, you see Chris and Megan here doing some of the processing. The capture of the images is relatively fast. It's the processing with different techniques to answer different questions that really starts to take a lot more time. But with that one capture, we can process the images in many different ways to work with different curatorial questions. So for the fragments that we've started to look at, this is a 14th century English fragment. And you can see here that from the color image, there's inks and there's some rubrication, some blues and some reds, both in the capital letters, but also throughout the page itself. So with some of the processing, there's a technique called principal component analysis. Essentially, that's a technique that allows us to detect and separate differences within large data sets. And I'll come back to another use of this further on in the presentation. So for example, with this, we create a false color image, as you can see there, because we want to see if the inks used in this are the same. As you can see to the left of that center image, it's sort of a bluey greeny tone, which relates more to the intensity, but they're all the same. If they were different inks, what we would start to see would be, for example, one would show up in the false color as perhaps blue, another might be red. So you can see the background here is very different to the ink because it's a different composition. So this allows us to start to understand whether there's different inks have been used, if there's annotations, has someone come along later or at a different time and changed that and added to that as part of the rendering of the text itself. One other technique we can do is false color infrared, where this allows us to process in a way that we can understand, particularly with historic parchment and manuscripts, is this a carbon-based ink or this an iron gall ink, which was used extensively historically. So we know from reference samples, as you can see here on the left, that iron gall ink will respond a reddy pinky color with the false color infrared processing, whereas other inks will respond black. So this allows us to see that the text and the annotations here are both an iron gall ink. Look at those colored images, those colored sections for the annotation, the capital letters and the colored letters. And here you can see when we do that principal component analysis and we process to look to see are those all the same or not, you can see at a quick glance that in fact all of the yellows look the same and the blues look the same. If those yellows were in fact slightly different composition, one might be orange and one might be yellow with the false color processing. So it allows us to very quickly identify whether they're the same or not. And then if we're going to look at those with some of the other techniques, we would know straight away that we need to look at more than one or two of these because some of them, even though they look yellow, are in fact different yellows. And you'll see more as I move further forward. We also are very interested in the construction, not just from the original material, but how it potentially may have been bound. So here you can see the low angle raking infrared drops out most of the ink, but also shows us indentations and markings from the original binding. This is where it gets really exciting because if we're looking at things that have been disbound and separated over the time of their life, this may be a way of seeing if those markings are exactly the same. Potentially, they could have come from the original volume. So there's so much information that we can start to pull out. It's just really exciting to work with all these different techniques. This is a Bible fragment from 14th century Italian, and you can see there some different scraping marks and some different lines that you're starting to see in the visible image there. We can see these under infrared and either scrape marks or potentially as they've pulled on some of the glue, potentially as that was made into a cover, as you can see from some of the markings there. So those striations, we start to look through the ink and pull up a better feel for whether that's original construction scrapings or removal of some other material at some point in the history of the lifetime of this document. And also we can start to see where the parchment was ruled prior to writing the text, which was historically either prick marks and metal point styli before they actually used a lead-based pencil there. And if there's enough of a response from what that is, we can use another technique to see whether that metal point is in fact silver or lead or some other component as well, which may actually relate to the composition of the actual text itself or where it was actually created. Another fragment here, a sacrament fragment from 13th century. As we start to look at this, when we do some of that image processing, we can start to pull out some of that faded or obscured information. This is actually just a black and white rendition of that, but what we can do then with the color processing is, as I said earlier, create a false color image, which very clearly separates different colored inks, but also starts to pull out some of those different areas where it's a little bit fuzzy from staining. The reason we often do multiple versions of the different false color combination is that different scholars look at, and all of us have different eyes. Some of them find different combinations easier to read. So we often work with specific scholars with different color combinations to make that the optimal visual reference for them. I want to move on now to another technique, micro X-ray fluorescent spectroscopy. Here you can see Cindy Conley-Ryan starting to look at this with one of our mapping XRFs, the Artex. And this actually is a non-contact way of looking at the trace metals in particularly pigments and inks, but also on the background of the parchment itself in terms of understanding how that was actually processed and created. As you saw earlier, Cindy's one of our staff who works extensively with creating historic recipes, and these really help us understand what the material sources are for these and how they've been prepared. So for example, with the vitriols, which is part of the Iron Gall ink creation, if they've come from different mines in different countries, they're going to contain different amounts of trace metals, whether that's copper, zinc, or other trace elements in the inks themselves. So you'll start to see that in some of the spectra as we go forward. So as you can see here, these two 14th century fragments that we're looking at, that Cindy's been looking at, and they've got markedly different vitriols. So if you look at the one on the left there, the spectra matches to a very intense peak for copper, the Cu peak there, whereas the one on the right, which if you were just glancing at in terms of the hand looks fairly similar, the ink from that one is actually very high in sulfur and manganese and a lot higher in zinc. So you see how we are pulling out a lot more information than you can see visibly when you look at an object. We're starting to really build up the knowledge and think about, gosh, maybe it's German. There's a lot of zinc in some of the German inks in mine. So we're really starting to get a feel for understanding where these collection items might have come from and what they can tell us from these techniques that they can't from just one look. Here's a 14th century English document, which is being, you can see on the left, transmitted light there to sort of, one, to understand what's underneath, but also because the technique we have, the beam will look through the entire parchment leaf. So we always make sure that when we take a spot, we're looking in an area where there's not something on the back. So we know that the signal we're getting is from that specific ink or that specific area of the parchment and not pulling in some other background information. This was a particular interest for these, where there's a lot of sometimes paper attachment from it being reused or adhesive. And so we want to make sure that the spectra we're getting is the pure component that we want to analyze. Here is another interesting component. So as I've been saying, you can't necessarily predict the composition from the visual appearance. This ink looks somewhat silvery, but in fact it's not metal leaf, but a lead sulfide. So that's an alteration product of what might have been potentially an original red ink, like red lead. So we really do need these techniques to actually build up that knowledge and understand what we're looking at. And it's also interesting then if you're trying to recreate what the original was, you know now that that was originally a faded red ink. And we're very interested, and scholars are always asking us, can we tell if the manuscript has one hand or several hands? And from that, we often are looking at the inks. There's also handwriting analysis, of course, but what can that document tell us about the ink composition from that? If you look at the image on the left, the center of the left-hand column there, if we compare the XRF spectra from the glosses and the decorative elements, we can see that that ink is the same for the main ink, the main text, as well as that drawing there. So that's interesting. Whereas we find that ink of the design region that's shown in blue on the spectra there, that has a distinct peak for lead, potentially from an underdrawing, and that comes through the other ink, whereas the other ink elements are comparable to the ink in the text area, and that's the spectra shown in red. So very often we sometimes create more questions than we necessarily answer, which is something that we're very happy about, but each piece of this information that we're gathering really starts to build the knowledge and the understanding of these materials. And some pigments can actually be identified through the characteristic elements. So as I said earlier, the imaging can tell us whether the blue is the same or not, but now we want to know exactly what we're looking at. So the 12th century fragment there from Italy, potentially a Great Bible fragment. The bright blue has aluminum, silica and sulfur, which leads us to an identification of ultramarine. We have another one which has lead white and then orpiment, potentially indigo, then a visually bright sample, copper and trace elements, leads us to understand azurite. So you see we're starting to actually extract more knowledge than we can from just visibly looking at, gosh, it's a blue pigment, and thinking what it might be. It may in fact be something that's not necessarily what we would expect from that time period or that country, which leads us to think about trade routes and other components as well. There's also regions on this legal text with these stains from water damage, and ink there has sort of a paler, redder hue than the surrounding lines of the text. What we're finding is that it matches perfectly with the surrounding darker ink, except that some of the calcium surface treatment from the parchment, as I said, we always do that background spectra of the parchment, looks to indicate that some of the calcium surface treatment of the parchment has been lost. So still building up that understanding of whether there's a difference in the ink or whether that's some sort of damage from fading or some other treatment or something that's happened in its lifetime to the materials that we're working with. Fiber optic reflectance spectroscopy is another technique that is used a lot for organic pigments. You can see Tama Viafana here capturing the spectra from one of the fragments. It's a very quick technique, which makes it something that we like to use, and it's very good at identifying pigments that are very similar visually, and also starting to distinguish between various binders that we have that are always used as part of the composition of different inks and pigments. So here we can see from the spectra on the right, we're starting to see two visually similar colors that are actually different. We've got azurite for the fragment 117, folio 3, and then lapis lazuli from the Greater Bible fragment. So we're building upon the knowledge that Cindy captured from that X-ray fluorescence in terms of the identification of the inorganic, the elemental parts that are there, and now looking at the visible and the organic unique spectra and adding that knowledge to that as well. You can also start to look at degraded metallic pigments as well. And this is a display leader, as Cindy had noted, was originally a brightly coloured red lead. So we can match this against a reference spectra and start to understand what's happening with that and do different derivatives and pull out information. As you can start to see from all of these different spectra, there's a lot of information and an incredible amount of expertise and work that our staff do to better understand, particularly what we're looking at as things degrade over time and what we're looking at and what might have caused that change over time. And again, here we're starting to look at different areas of the parchment that have different spectral responses. We want to look at some of the degradation pathways and understand why we're seeing different responses on different materials and what that might mean in terms of the history of that, but also how we can better preserve our collection materials as well. So you can see here from the center component for those regions of interest that we analysed, 2 of those are right on the edge where there'll be a lot more handling and would expect different oils and potentially more damage, versus an area more towards the center of the manuscript that potentially may have been handled less and have less damage. Another technique, Fourier transform infrared spectroscopy. As you can see here, another portable technique, and you'll see Hadley Johnson working in the Rare Books Special Collection reading room here, and starting to now pull up some more of the, again, organic information on the collection item itself. As you can see from the placement there, we can hover over the object and just bring it down to the surface, but not even touch the surface. So all of these techniques we've been talking about are non-invasive in terms of not coming up to, but not necessarily touching the object in any way, particularly if it's very fragile. And this is a technique, as I said, we're using the infrared light to absorb and reflect off the surface and start to identify some of the functional groups of those organic components, whether it's of the parchment itself or of the reference spectra or of the inks. And this helps characterize the organic components. So as we'll talk about a little bit more, we're very interested in what we call amide 1 and 2 bands. The amide is a component of a protein, and that is part of the collagen that the parchment is made from. And we're very interested in seeing how that shifts in relation to the condition of historic parchments, why some are in good condition and some not so good. So as you saw from the charm material earlier, PITT has a reference collection of parchments that represent a number of different species of animals and different preparation methods. Here you're seeing some where they've been bleached or natural. We also have others that have been processed by colleagues, Jesse Meyer from Pergamire, who have actually captured them at different aspects of historic processing, and that allows us to better understand whether it's the liming or other processes in the construction that may actually have been done slightly differently on the fragments, and that's why we're seeing different types of composition and condition. So as I was saying there, you can see here the different parchment responses from the surface in terms of the signal. Here we've got the Dare spectrum with both natural and bleached, and there's a tiny component there that we're seeing the calcium carbonate from the bleaching agent. Now we're comparing those reference spectra with some of the fragments, and what we're looking at is with those 2 amide bands, the intensity of them, if they're closer, and the height can actually indicate different levels of degradation. So as I said, we're still early in the stages of this. We're really delving into some of those, particularly this one on the upper right, where we've got potentially some heat damage regions on that, really starting to extract out what we're seeing and how our reference samples can help us analyze and extract from those components. And you're saying, you know, I'm looking at these spectra and they look pretty much the same. So Andrew Davis has been doing a lot of work in an area called chemometrics. It's looking, essentially that means chemistry statistics. So we're basically looking statistically at all of those different wave bands. Instead of spending hours looking at them by eye, we're now doing some processing, like that principal component analysis that we did with the imaging, to extract out how we can actually identify. We first developed this with predicted pulp for paper, and now we're working to use the reference materials to characterize the species. So again, we're using some specific peaks from the infrared that you saw from Hadley's work, and Andrew's been using those identifying peaks with the statistical analysis, and you can see on the left there a very distinct grouping for calf, sheep, goat and deer. It's very early stages. We will keep building this up as we get more reference samples and more positive identifications of that. What you're seeing now is we've taken some of those fragments and put them into the model. So we're seeing one of them that looks to be potentially goat, the English potentially calf, and the Greater Bible fragment, it's a bit of an outlier. It's giving us challenges. That may be that there's been some of the damage or some of the processing or some glue or some other component is masking what the infrared can see. So we continue to build up the model to understand more and be able to use these non-invasive techniques to characterize the materials and take that information back to the collections. And you can see here just another version of that. We've got sort of the training model on the left and then the validation on the right there. Another technique that is slightly invasive came from discussions in the Books to Beasts collaboration we work with international colleagues, where conservators sometimes do a little bit of a razor rubbing to clean. And the thought was, from Sarah Fitterment and Matthew Collins and others, could we use this conservation accepted practice to capture something from the eraser rubbing? So you can see here Chris Bolser doing a very gentle eraser rubbing from an area to capture some of the, because the eraser absorbs some of the parchment information, and we can use a technique called matrix assisted laser desorption. Ionized time of flight. No, you don't have to memorize that. It's a mass spectrometry technique. It's essentially fingerprints those organic materials. In this case, helps us identify the particular species. So we can start to build up knowledge and validate and verify the chemometrics as well from this technique. Here again, you can see just a gentle razor rubbing from there. Two more techniques before we wrap up and open for questions. It may seem unusual, but we are very interested in what the volatiles profile of different objects is, what it smells like, because that tells us a lot about what is off-gassing. Here you're seeing Kelly Stoneburner actually creating a special box with Eric Munro to capture from a parchment. And from this, we start to get a lot of different materials and different peaks coming off from the mass spectrometry there. And from that, we can start to detect materials, for example, whether we're seeing fatty acids that might come from the parchment, paper, we might see acetic acid, a lot of things that either we can see from the surrounding materials. So we often do use this technique for housing materials and building materials, but also to build up a profile. And this adds extra information for us to understand what might be happening in terms of the degradation. And if we're not seeing off-gassing, knowing that it's actually in very good condition. One of our new techniques that has been very interesting is 3D imaging. You're seeing here Eric Munro doing a couple of versions of capture from the surface of a parchment. We can take this and do a handheld rendering, or we can also put it on a tripod and automate it somewhat, which is a lot easier. From the image on the left, you're starting to see there that there's tiny little dots around this, little similar to some of the reflectance transformance imaging some of you may be familiar with, where we use those markers for the 3D imager to have a good idea of where it is. So to understand as it scans and adds extra components of the parchment as it does the scan, what are we actually looking at and how does it overlay and make sure that as it goes to a new area, it's matching up and mapping all of those areas together. This is particularly important if there are large areas where there's no text on the document. It needs those fiduciary points to map up and capture that. So you can see here we can use a 3D table, which is gives us even more information and more control and less need to actually move the object or touch the object in any way. Here you can start to see that we reconstruct the material. What's very interesting and what I wanted to point out is we have an object where it's not 2D. We often think of paper and parchment and manuscripts as 2D. They're in fact 3D. Now we can actually measure the height of the difference in those cockled areas and understand and start to get a feel for the topography and what that might mean in terms of preservation or for conservation treatments and other areas of that as well. So you can see here as we look across the parchment, the average is about 1.5 millimeters deep in the troughs there, and these can be made at any point across the object. So we don't need a caliper to actually touch the object. We can really get a good feel for what's happening, how repetitive or different these measurements are. So this is something we're really starting to explore and add to the information we know from some of these learning from these materials. And just because it's just such a lovely way of thinking about the material, just want to show you again here the way we can start to rotate and look at these collection items and zoom in and really get a feel for what that might look like and layer these over the other techniques. So the future of the fragments, as we start to compare these fragments with reference parchments, with inks, with pigments and other manuscript documents, we're really starting to connect some historic construction techniques, and from that composition and characterization of the materials, whether it's looking at the species of the parchment or the inks and understanding whether they're faded or not, or have different metals, trace metals in them, and therefore might link to different locations or different scribes. We're starting to build up and connect collection links between the items themselves, both in our collections and in others. And this to me is incredibly exciting because we're really building our knowledge and we're expanding outside our collection, linking our collection with other collections, and also adding on how we can expand and know what condition they're in and work with our amazing conservation staff to expand that knowledge as well. So I'm going to finish there. I want to thank you for listening and open up for questions.
Thank you so much, Fenella. That was really amazing. We have one question to start with. And just a reminder, if you could enter your questions into the Q&A, it's easier for me to see them there. All right, so our first question is, what resources do you use to recreate historic inks? And do you use primary or original source recipes or analyze the inks and work backward, or some combination?
Fantastic question, thank you. So as I mentioned, Cindy has been and others have been doing a lot of work with creating from original recipes. As some of you may know, that can be a bit challenging because sometimes it's not very complete. It's like when you get a recipe from your grandmother. She says, well, just add a bit of this, a little amount of that. So some of it's like, well, what is a little bit of this? Cindy grows things in her garden. I'm still trying to talk Jake into actually creating a pigment garden at the Library of Congress, which would be fantastic. But we do that from original sources, some locally, some internationally. Some of those we do buy from pigment locations. But as you know, many of the modern pigments may look like, for example, indigo, but are not necessarily made from indigo. So a lot of that is trial and error, as you saw from some of those images of trying to do that. We also then, as to your point, work back from what we're collecting from the collection item. For example, on another project, we were seeing some odd anomalies. So we then started to create compositions of both iron gall ink and carbon, rather than just working with one or the other.
Thanks. Have you been surprised at results of the analytical methods for parchment source species identification versus what was expected using only visual examination?
That has been a really interesting area. And yes, sometimes it has. A lot of people have looked, for example, and conservators are very good at doing this, but sometimes they have been surprised when the follicles didn't necessarily match what was expected. And this has been very interesting. And as I said, with the Books to Beast project, getting some very unique responses. And so, yes, it has been quite surprising. What we've found has also been wonderful when it's verified what people have thought, but it's made us start to think a little bit more about how we want to add in a technique that can explore and expand and really confirm definitively what we know about that.
Are there any analytical techniques that can be used with a digital camera, perhaps with a bit of retrofitting?
That's a great question, and I hope we will be doing Megan Wilson and Chris and I have done some work with that. So essentially, yes, there is. I won't have time to go into that in depth here, but you can essentially, from your RGB camera, you can add in, for example, one UV light, one IR if you modify and take out the IR filter. Essentially, even with 5 bands, you can capture quite a lot of information from a modified camera. And we'll be actually talking about that at Imaging Science and Technology Archiving Conference.
That also sounds like a really great webinar for us in the future too. Okay, so someone was wondering how these fragments are stored, especially those detached from its original text. Is that something that you can answer, Fenella?
I'll talk a little bit to it, but as I said, that would be another wonderful webinar from Mariana Stell, and conservation staff. So sometimes fragments come in as part of a special collection, and there's so much work to do. Right now, they are being carefully separated with safe sleeves and with as much information as can be added to that. So they are being separated from each other, and they're in, as I said, the unique housing material approved plastic sleeves to separate and keep them apart from each other. And I forget, I think there was another part to that question that I may not have answered.
Especially when they're detached from the original, is there any sort of special way that they keep track of that?
That's where, part of the work we're doing in this fragmentology research is trying to add that analytical and the curatorial knowledge to start to look at, for example, there are 3 that are from the same from a great Bible, and we're looking very carefully to see if, in fact, those 3 are intimately related and which point they would be housed together to keep them as one series of fragments, so to speak.
Okay. And somebody asked about the video recording. I'm working on that answer in the chat box. Okay, what is the relationship between this kind of research and the paleographical and textual researches?
So there's sort of connections, and it depends very much on the various collaborations that we're working on. I probably don't have time to go into some of that, but we work to interconnect those collaborations and those different sources of knowledge, because as you've seen, everyone has a different expertise, and we could look simply from the analytical perspective and we would see this is different to that, but without, for example, Mariana's looking at and saying this is clearly English because of X, or this text layout means this, that's where that curatorial and scholarly knowledge adds. So it's a very iterative process, and all of us work to expand and connect and add those different expertises together.
Are you aware of any further testing being done beyond parchment speciation from the collected eraser crumbs, maybe DNA testing?
Yes, there is, and I don't have a chance to put that up, but maybe we can get that. The Books to Beast Serum Firmament is doing a lot more work with the DNA. The reason we've done a little bit of work with some of that, it takes a lot more sample, and it's still being worked on. We are working on a technique in-house to be able to do that with some of the Māori and some other LC techniques to be able to do that speciation in-house. So that's something we're working on within PRTD. But there is other work for the DNA as well. They are looking at different preservation or liming indexes to try to get a feel for the degradation aspect. The challenge is that they need to have a good provenance of whether it's age or whether it's part of the processing, and you need to know the original construction, which is why we're doing the reference samples to tease out what different processing techniques might look like, in the after they've aged in various ways.
And someone else would like to know how the pandemic impacted various aspects of your work.
So we, as you can see with the imaging, for example, and with a lot of these techniques, we're doing a lot of work with big data. So we're collecting a lot of data. We were fortunate just the week before the pandemic, I collected a lot of hard drives. Staff went home with hard drives crammed full of data, and we shared hard drives and we shared portable laptops with some of the different processing software on them. And I just want to say a huge kudos to all of the Preservation Directorate and our support from Jake and also my staff who worked non-stop throughout the pandemic. So while we couldn't be in the lab, we did in fact manage to do extensive amount of processing of different data. We developed a lot of the chemometrics modeling. We did a lot of visualization of data, and we really, and a lot of publishing of publications that have been languishing. So while it was challenging because we couldn't be in the lab, I just want to say a huge thank you and incredible work from my staff who worked 100% throughout the pandemic with pulling a lot more out of the information from some of the pre-processing and the projects that we had captured prior to the pandemic.
I also noticed that there is an Art Chimes with their hand raised. If you want to ask your question out loud, I can let you do that. Just let me know in the chat box. Okay, and we have one more question. This is actually from okay, this is actually from the music division. They have several musical fragments dating from the 10th through 16th centuries, and they have gathered them and digitized them. And they were wondering if there is more information that they might discern from your type of testing.
Absolutely. So we would be able to see if what the paper or the parchment was, what the composition of that, whether the inks, particularly if they're looking at the same composer, is it the same ink or not? And so all of the techniques that we've used on this can be applied to those. So please feel free to reach out to me, and as I said, we'd started with Rare Box and Special Collections. We'd love to work with you on that as well.
Well, I think that wraps up our questions for today. This was such a fantastic presentation.
Oh, thank you, Nellie, and thank you all for listening. It's just so wonderful being able to share some of the work we do, and this was particularly learning from our collection is just so exciting.